So let's talk more specifically about the different pathways. So either Ficalin or MBL is going to come with a MBL is mannose binding lectin, and Ficalin's also recognized carbohydrates, but a different carbohydrate than lectin, um, are going to come with the MASP or MBL associated serine protease. Serine proteases are able to cut proteins at serines. And so they are going to cut C4 and C2. When they cut C4 and C2, those two proteins are called the C3 convertase. Those two proteins together are called the C3 convertase, and they cut C3. When you cut C4, C2, C3, the two pieces both do something. So in the case of C4 and C3, um, the the smaller part, the C3A or C4A, are anaphylatoxins. They cause inflammation. In the case of C2, it also causes activation of other pathways that are less understood. So in the lectin pathway, it is going to be the recognition of sugars that people don't have, but bacteria do, fungus do, and that recognition is going to be enough to cause these um, um, Ficalin and excuse me, MBL associated serine proteases to arise. Um, in the case of the classical pathway, um, the C1q interacts with antibodies that are on the surface of the protein. So we've decorated the surface of the pathogen. So we've decorated the surface of the pathogen with antibodies, and that recruits complement. Um, so C1q shows up with uh, C1R and C1S, and they act as serine proteases also. So so they're also going to cut C2 and C4. In the case of the alternative pathway, um, it's going to be the spontaneous hydrolysis of C3 that initiates the deposition of C3 onto the surface, and then factor B shows up, binds to it. Factor D cuts, um, and you end up with a C3B BB complex, and that in conjunction with piperidin uh, um, acts as a C3 convertase. So once you have a C3 convertase by whichever means, um, lectin pathway, classical pathway, or alternative pathway, now you're producing a whole bunch of C3. And that C3 deposits itself on the pathogen's surface, and that's because it has thioester bond inside of it. And so you break that thioester bond and you create a covalent linkage between the carbon that was bound to the thio group, the sulfur, and that carbon is very uh, reactive and can cause a covalent bond to the surface of the bacteria. And that C3b now is able to act as a to do proteolysis of C5. Um, so some of the stuff I just talked about, the deposition of the C3 is because of the thioester domain inside of the C3b, once it gets exposed, it can now cause a covalent linkage uh, of that domain onto the surface of the pathogen. Um, that um, that is a reversible reaction, and so hydrolysis would break the C3b bond to the surface. Um, so so in all three cases, lectin, classical, and alternative, once you've got a C3 convertase, you can create a C5 convertase. So in the lectin pathway, the C3 convertase, and the classical pathway, the C3 convertase, in both cases, is C4B2A. In the alternative pathway, it's C3BBB. And so once you've got that C3 convertase, they only have to cut one C3 to create the C4B2A3B or the C3BBB. Um, so if you've got two C3Bs and a BB, then that'll be a C5 convertase. And if you've got a C4B2A3B, then that will be a C5 convertase. So, so the lectin pathway is because vertebrates have a very specific uh, um, glycoprotein structure, where we have fucoses and sialic acids. And in the case of yeast, really they've got this like mannose, and that's what they've got. They've got just this large mannose complex. In the case of bacteria, you've got a mannose and glucose. We talked about that just a second ago in video 2, what their glycoproteins look like. Um, but uh, but they look different, and because of that, they can be recognized, and they can be recognized by either MBL or phycolin. Phycolin and MBL have very similar structures in that they've got collagen helices that bind to each other. That collagen helix binds to an alpha helix, um, and so then those will bind to each other. And so the only difference between MBL and phycolin is their carbohydrate recognition. Either they're going to recognize these sugars or they're going to recognize fibronectin. In either case, they're going to bring a MBL associated serine protease with them, and that will act as the uh, the protein that does proteolysis of first C4b and then C2, I'm sorry, C4 into C4a and C4b. And remember, C4a is an anaphylatoxin and causes inflammation, whereas C4b just drifts down to the surface. And then C2, which C2b goes off and does stuff, and C2a binds to C4b. And those two together are a C3 convertase and can make many, many, many C3bs, which are going to bind to the surface through a thioester bond. C3a is also an anaphylatoxin and it's going to cause inflammation. So we're going to end up with a lot of C3bs on the surface. This is going to do several things. One is that C4B2A3B is a C5 convertase, and C3b can get recognized by phagocytic cells. In the classical pathway, on the other hand, we are going to initiate it by antibodies, but you can see how the structure ends up being very, very similar. You still got that collagen region to bring the pieces of the C1 together. and then associated with the C1Q will be C1R and C1S, which are both going to act like MASPs. They're still serine proteases. So the C1R and C1S are then going to cut C4 and C2. So as far as anaphylatoxins are concerned, the weakest anaphylatoxin is the C4A. C3A works a little bit better, and C5A works the best. And then, like I said, C2B goes on to be vasoactive also, but not it's not going to act like an anaphylatoxin and produce all of inflammation. So what are we going to do once we get the C3B on the surface? Well, C3B is going to cut C5, and so that's going to be an important next step. The alternative pathway, like I said, it's the older pathway, and it starts by spontaneous hydrolysis of C3, which recruits factor B, and then there will be proteolysis to create C3B, and the C3BBB is going to act as a C3B convertase. And so then once you've got a C3BBB complex, that will recruit factor P, and that will act as a C5 convertase. We also have inhibitors, and so anything that you turn on, you want to be able to turn off. And so CR1, factor H, DAF, MCP, they all act as inhibitors, and the way they do that is by recruiting factor I. Factor I then inhibits C3B, and so it can no longer act as a C5 convertase. So anything you turn on, you want to be able to turn off. So what's the purpose of creating these protein complexes is to create a C5 convertase. Besides that, C3 is going to increase phagocytosis. That's also true. But once we get a C5 cut, C5A is the most potent anaphylatoxin, and C5B then goes on to create the membrane attack complex. So C3B by itself will not cause phagocytosis. It will get bound to the surface of a phagocytic cell, but we also need some amount of C5A to complete the signaling and cause the phagocytosis. So C3B goes on to cut C5, and then the C5A is drifting around, and it binds to the C5A receptor and causes phagocytosis. So phagocytosis is one of the parts of complement. Another important part of complement is the membrane attack complex, and that works by C5B then recruits C6, C7. They drift down to the membrane surface, and then C8, and they recruit C8, and C8 goes into the membrane and recruits C9. And then you recruit a whole bunch of C9s, which create a pore. And this is a really big pore. Look at that arrow pointing at that pore. This is the bacterial cell, and see that protein complex there? That's a straw that's going to cause the guts of the cell to just burst out of the cell. So that's the point of complement, is to create the membrane attack complex. It does help with phagocytosis, but the membrane attack complex is really the most potent effector. Okay, so that's it for chapter 2.